Good morning and Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity. My name is Wahyu Widayat. I am from Department of Biotechnology Faculty of Postgraduate Padjadjaran University, Bandung, Indonesia. Okay, I want to present my paper result. With title is Immunoinformatics Study in Prediction Cytotoxic T Lymphocyte Epitope of Sars-Cov-2 Spike Protein. Okay, first introduction is the pandemic outbreak that occurred at the end of December 2019 changed the behavior of all human beings in the world without exception, known as the new normal. The first case was reported in Wuhan and quickly spread to various countries, including Asian countries. World Health Organization announced that the pandemic was caused by a novel coronavirus originating in bed, later confirmed as SARS-CoV-2 and now as COVID-19. In early March 2020, the virus was confirmed to have spread in Indonesia and spread uncontrollably to various regions. Efforts to develop an effective vaccine have been initiated in many countries, including Indonesia. New vaccine types include DNA, viral vector, recombinant protein, adenovirus, mRNA, and peptide-based vaccine. It stimulates independent from vaccine, especially in the current situation against SARS-CoV-2. An epitope is a peptide defined as a group of amino acids from protein antigen that interact with antibody or T-cell receptor, thereby activating an immune response. T-cell epitopes are expressed by major histone coupled complex protein or in human known as human leukocyte antigen. T-cell-based cellular immunity is essential to fight SARS-CoV infection in patients suffering from COVID-19 of varying disease severity and at various post-infection stages. They found SARS-CoV-2 specific memory T cell in most recovered individuals, including asymptomatic case. We target the spike protein as a source of candidate antigen epitope. The spike protein undergoes of conformational chain to carry out membrane fusion by interacting with the cell surface angiotensin converter engine 2 receptor and generate humoral and cellular immune response. Several vaccine platforms such as Moderna, Pfizer, and AstraZeneca use spike protein as an antigenic source resulting in more 90% adequate protection. The rapid development of analysis of the pandemic genome to identify antigenic protein and discovery of new vaccine using reverse vaccinology were rapidly carried out to informatics. It took immunoinformatics, which would reduce the time and cost required for laboratory analysis. Currently, the study aims to predict the cytotoxic T-cell epitope of the SARS-CoV-2 spike protein using an immunoinformatic approach as a vaccine candidate. Several informatics software used were to explore spike protein to screen for many potential epitopes. We hope that the finding of this study will contribute to help toward the development of multi-epitope vaccine against SARS-CoV-2 population in Asia, especially Indonesia. Okay, material method is spike protein sequencing protocol. The full DNA sequence of SARS-CoV-2 Indonesia was taken from the GSAT database with the accession number APAASL 5299967. Then it was translated using the Expressy web server. Then spike protein sequence was selected and saved in FASTA format. And next is a prediction of cytotoxic T lymphocyte epitope against HLA class 1. The prediction of epitope using net MHC pen 4.1 software to predict binding affinity and percentile rank across all representative allele of each HLA class 1 on supertype of human leukocyte antigen HLA-A and B present in Indonesia population. A strong binder prediction score in chosen 0.5%, with the top rank will be selected as the epitope. Next is prediction of toxicity and allergenity of cytotoxic T lymphocyte epitope. For the toxicity, we use the ToxinPred software, and the allergenity was predicted using Allertop 2.0 software. Next is autoimmune and conservation analysis. The concept epitope was analyzed using conservation analysis on ADB server with a 19 until 100% identity value criteria. Meanwhile, autoimmune analysis was performed using the BLAST program protein in NCBI to find homologous sequence between epitope and human protein. And the last is the cytotoxic T lymphocyte epitope modeling against HLA class 1. The CTL epitope modeling of spike protein was carried out using model 10 application. The model was selected based on the lowest second optimized protein energy value and visualized using the BioVa discovery software. Quality modeling will be seen on the web server project to see the result of the Ramachandra report. Okay, the session about result and discussion. Okay, the figure 1. This is the figure 1. Figure 1 shown the sequence Indonesian spike protein SARS-CoV-2, and figure 2 is alignment sequence Indonesian spike protein SARS-CoV-2 against Wuhan human. Okay, the sequence that we have a transfer from expression. Then we do the alignment for the first human sequence. And the result is that there is a mutation in residue SIG14 where aspartic acid convert to glycine, or also called variant D SIG14G. The mutation significantly lower in Asia countries, but strong correlation with increase of viral load and exhibit a strong affinity to binding to S2 receptor. So there is possibility to increase infectivity. So this is the reason we started for work using local spike sequence. Okay, after we get the sequence from Indonesia, we prediction of the cytotoxic T lymphocyte using net MHC pen 4.1, and we select 16 epitope at HLA-A 2L1, 24 at HLA-A 11L1, and 8 epitope HLA-B 40L1. So the table described for each of epitope from supertype HLA. The reason okay, not the reason, but we chosen 9-mer peptide line to predict because the line distribution of naturally processed and present peptide in generally 9-mer peptide. So the prominent Indonesian human leukocyte antigen, namely HLA-2L1, HLA-11L1, and HLA-B40 L1, especially in Japanese and Sundanese and is also present in the population of countries in Asia. Okay. After we obtain the result of the net MHC1 prediction, we process with the toxicity and allergy. Okay. Figure 3 explain all the epitope predicted by toxin threat are not a poison. So our epitope is not a poison. And table 2 explain that allertopedic 10 epitope on HLA-A 2.1, HLA-A 12.1, 11 on HLA 11 L1, and 5 on HLA B40 L1, not as allergen. So the epitope have amino acid similarity with organism sourced from Solanum tuberosorum and Trichum iscitivum is wheat, and Origata viva is rice, and Homo sapiens is human. And next is the last of prediction safety from epitope. Non-toxic and allergen epitope are then analyzed for autoimmune and concept analyzed. We'll see the table 3 describe the non-autoimmune and concept epitope. We obtain two epitope from each HLA. We analyze concept epitope of 687 Indonesia SARS-CoV-2 spike protein sequence, and we choose the epitope which has 100% concept. We also examine the novel of epitope on the IDD server database. The result that 5, 1, 2, 3, 4, 5 where's a new epitope or not previously published. Can you see the highlight yellow color is a new epitope? 1, 2, 3, 4, 5, 6, 5. Okay, the last is modeling cytotoxic lymphocyte of epitope. So we model the new. We focus for a new epitope, yeah, for the model of 3D protein. The model selection is based on the lowest dose value. The structure of the model show high stability and is close to the original structure if it has low dose value. Okay. Figure 4 is 3D structure and Ramachandran plot epitope CTL from supertype HLA. So there is shown the model has the model have a good quality. The quality in profiling and overview of stereochemia, structure of the interaction between the epitope and its HLA supertype. So a good structure is a structure that has a presence of the most favorite region greater than 50%. The greater percent of the acid residue is the most favorite region, and the lower percent of the residue disallowed region of structure will be good quality. So overall, it is model is described to a good model or good structure of leukemia because this solar region because this our region is low and most favorite region in 93% until 96%. Okay, the next is conclusion. Based on the result of this study, we conclude that we obtain the 6 potential epitopes of SARS-CoV-2 spike protein from each of the SLA A and B subtype in Indonesia, and 5 of them were new epitopes. We realized that this research is primarily study and there are still many weaknesses, so it's necessary carry out validation and laboratory experiment. We hope that our result provide an overview of potential epitopes of SARS-CoV-2 as a vaccine candidate. Okay, thank you for the attention and terima kasih.